Thank you for joining me for our second and final eReport in order to discuss the latter material of this course, Cellular Biology. This is a great opportunity to tell the story of the cell through the lenses of its communication, transportation, and formation of communities in order to understand the bare necessities of life itself. Without further ado, let's discuss the cell membrane. The cellular membrane is a thin, fatty film layer that separates the cell from its outside environment. The membrane functions as a protective gatekeeper, as you can see, in order to maintain the biochemistry of life by fulfilling the functions of both a portal and a barrier. This is accomplished by membrane proteins such as pumps, channels, or receptors that monitor cellular signals. In this way, the membrane can accomplish a number of large feats, including receiving information, controlling the import and export of material, and maintaining the capability for movement or expansion of the cell, and within the cell itself. And I thought that this funny picture was representative of the fierce and proactive nature of all of our cell membranes, which are both regulatory and fluid in their functions. So the cell membrane is actually a mechanical marvel. As a phospholipid bilayer, it spontaneously shapes, flexes, and grows itself in response to both tearing and movement. As we can see from this image here on the left, the phospholipid bilayer is comprised of two opposing layers made up of hydrophilic heads and hydrophobic tails that mirror one another. Because lipids are insoluble, membranes can block diffusion of water-loving molecules by way of these hydrophilic heads that line up side by side so closely. Hydrophilic and hydrophobic molecules are ambipathic, meaning that they spontaneously form membranes due to their polarity and opposing affinities for water. Instead of forming these cells, which we see here on the right, where hydrophobic tails huddle together and face one another, liposomes are instead formed spontaneously because hydrophobic tails of a bilayer are too bulky to form a micelle. This is the liposome here. And as you can see, actually, some of the hydrophobic tails are kinked, and it's because of the shape of the tails that makes it so that they cannot form micelles and instead form liposomes. And the only way that the form of a liposome's finite sheet can have no free, exposed edges is if it forms a self-sealing ball, where all of the cell's contents are actually enclosed within. Hence, we can then understand the spontaneity of a cell's membrane structure. This is energetically favorable for the cell because as a self-sealing membrane, it's conserving its energy through the careful regulation of biochemical balance. So pictured here, as we can see, is actually the fluid structure of the membrane. Although this structure may look rigid in some ways like we described, individual phospholipid components can actually laterally diffuse or float, or transversely diffuse and flip in order to change its shape while still maintaining the tight sealed barrier. Flipases are the enzymes that are responsible for restoring the shape of the lipid bilayer through this movement. A cell can actually modify its membrane's fluidity by regulating the length and double bonding going on between lipids, but this is not actually the case in eukaryotic cells. And instead, in eukaryotic cells, the presence or absence of cholesterol, pictured here in between the phospholipid bilayer, modifies the space in between the kinks of the hydrophobic tails in order to stiffen and loosen the membrane's integrity. And the membrane is asymmetrical, meaning that the protein composition of the membrane differs from inside to the outside of the cell. As you can see, there are some proteins present on the extracellular fluid side of the cell membrane that are not on the inside where the cytoplasm is. And this is to allow for the mediation of transportation across the cell membrane. So the proteins along the membrane that differ from the inside and outside actually confer specific function, and different organelles have different types of membranes. Membrane proteins allow for cross-membrane transportation, the anchoring of receptors, or for enzymes to bind. These are versatile functions that are all mediated by the membrane structure. Proteins can be embedded, literally stuck in the membrane, like this one, also known as integral proteins, or they could be peripherally attached covalently or non-covalently to other proteins that are themselves anchored or embedded to the membrane. Some of these membrane proteins are called transmembrane proteins, like I pointed out earlier, which have membrane domains that are embedded within the lipid bilayer. This restricts the movement of the membrane and prevents the floating of the membrane that we discussed earlier. Glycolipids, pictured here, have sugars attached to hydrophilic heads on the membrane made of oligosaccharides, or multiple individual sugars, on the exterior face of the membrane. The carbohydrate membrane layer provided by glycolipids allows for damage prevention to the membrane, cellular recognition for particles associating with the membrane, and adhesion onto the membrane for those molecules, which adds to its well-rounded functions. This is accomplished through the binding of lectins to the sugars. The membrane itself is supported by a meshwork of proteins called the cell cortex, which lies underneath here and within the cell in the cytoplasm. Water-loving or hydrophilic polar charged molecules are blocked from passively diffusing across the cell membrane because of the structure we discussed. But the movement of these molecules is actually pivotal to the many functions of life we have already discussed in cell and molecular biology. So this begs the question, how does the cell ensure that this movement happens? And the simple answer is through the collaborative system of membrane transport proteins, and there are many of them. Embedded or integral proteins are highly selective, but have the ability to bypass the limitations of the cell membrane. Of these, there are transporters that have moving parts in order to change shape in response to these molecules under certain conditions, depending on the type. Usually, transporters transport only in one direction. Channels are protein pores with no moving parts that are selective to specific ions as they pass down their concentration gradient. The ion concentrations on the outside and the inside of a cell differ intentionally. Their movement and transport is pivotal to cell activity. The balance of the electrical charge at the cell membrane as a result of this ionic difference equates to the structural integrity of the cell, and this is because localized and minor chemical imbalances can be controlled and regulated at the membrane. As you can see from this diagram, it's just demonstrating the ionic differences from the outside of the cell to the inside and the many ways that the ionic balances can be mediated or changed through either passive transport or active transport. Charged ions are mediated by membrane transport proteins known as ion channels or pumps, as you can see here, which are marked as exclusive for that particular ion it is custom to. The charge and the size of the molecules determine what kind of transport protein is ultimately required. These can be divided into passive and active transportation through proteins. There are passive channels and passive transporters that facilitate diffusion, and active transporters that facilitate active transport, which requires energy, or ATP. Even organelles within a cell have their own transport proteins, as each organelle has special biochemical needs, such as the mitochondria. Channel-mediated transportation results in the passing of ions through a pore that corresponds with the concentration gradient of the ion it is selective to, as I mentioned in the previous slide. Carrier-mediated passive transport is characterized by a shape change in response to a particular molecule. As you can see, this is the shape of the receptor here, and once this molecule binds, that shape changes in order to allow it to insert itself into the cell membrane and through. Coupled transporters are used for when a molecule is transported with its concentration gradient along with another molecule that requires movement through the transporter. These can be symporters, which ensure the movement of the coupled transporter ions in the same direction, or antiporters, in which the movement of one ion down its concentration gradient allows for opposing movement of a molecule in the opposite direction. This is directed by conformational change, and coupled transporters can either be symporters or antiporters. In active transport, the membrane transport proteins are controlled by ATP, as I mentioned, in order to move the molecules against its concentration gradient. The sodium-potassium pump is the perfect example of an active transport protein that is needed to sustain cellular function. It requires approximately 30% of a cell's ATP energy in order to maintain the balance of sodium and calcium ions between the interior and exterior of the cell. You might say, well, that's a whole lot of energy for one transport protein to use for us. But in reality, the job of the sodium-potassium pump is pivotal to our cell's survival and therefore our survival. The sodium-potassium pump is important for maintaining the low concentration of sodium ions within the cell membrane to prevent the bursting of the cell. And it is through these specialized active transport proteins that we can control our osmotic pressure in all of our cells, as too much sodium would cause the cells to swell. So as we discuss the transportation of molecules across the cell membrane through transport proteins, the budding and the fusing of transport vesicles is responsible for all of the transportation of materials throughout the endomembrane system in our cells. This has been organized to ensure that all hydrophilic proteins can be properly packaged and transported throughout the intracellular components of the cell and outwards out of the cell. There are two pathways by which vesicular transportation can occur. The secretory pathway, which occurs from the endoplasmic reticulum to the Golgi and outwards to the cell membrane, and the endocytic pathway, which is the transportation from the cell membrane to the innermost parts of the endomembrane system. Pictured here in entirety is the process of a vesicle gathering its cargo, pinching itself off, budding off the membrane, and traveling, uncoating itself, tethering, and fusing with another membrane. It truly is a brilliant system in order to ensure that our protected organelles can each selectively receive the required proteins in order to fulfill their cellular function. Vesicles are coated by a special protein called clathrin, which I like that name very much, which comprises the cage-like membrane that encompasses the vesicle wholly as it is formed. Every vesicle begins as a clathrin-coated pit, which surrounds the pit in a basket-like structure, like we can see here in the dark green. And every vesicle begins this way in order to begin the pulling off the vesicle from the membrane itself. A vesicle is formed and pinched off into its clathrin coat with the aid of dynamin, which actually restricts and forms the ring around the neck of the vesicle in order to pinch it off. And although clathrin provides the primary support for the formation of the vesicle through its basket-like structure, it is the protein adaptin, seen here in light green, that is ultimately responsible for ensuring that the correct cargo is included inside the vesicle itself while it is being formed by clathrin and dynamin. This is accomplished by certain subtypes of adaptins that associate and select certain cargo based on the specific transmembrane protein signals that are associated with that subtype of adaptin and the cargo itself. Adaptins collect the cargo into the vesicle in order to ensure that it is properly packaged prior to dynamin pinching off the membrane. And once a vesicle is then pinched off and coated completely by clathrin and then filled with its cargo, it is destined for a destination through the inner workings of the endomembrane system, ready to fuse with its destination membrane. Once a vesicle has reached the destination membrane, the Rab proteins, which we can actually see right here in the right half of this image, are specialized for that particular membrane and associate with the vesicle in order to prepare the vesicle for its fusion. These Rab proteins have subtypes that act as a matchmaker between the target vesicle and its target membrane to ensure that the wrong cargo doesn't go to the wrong place. A Rab protein will then be recognized by what is called a tethering protein, or here it's called a tethering factor. And the tethering proteins fully associate with the vesicle with the membrane by pulling it closer to it. There is another protein called snare proteins that actually ensnare the vesicles in a way by pulling the vesicle even closer to the membrane and docking it for fusion so that it's prepared to unload its cargo when it receives the signal to do so. So basically it holds it there until it is prepared to completely fuse with the membrane. There are V-snare proteins, which are located on the vesicle itself, and then there are T-snare proteins, which are located on the membrane the vesicle is docking to. These two snare proteins intertwine one another, pulling each other closer, and when the time is right, the snare proteins will twist and twist until the vesicle fully fuses with the membrane, releasing its contents that were within. So I think this is one of my favorite topics by far, the cytoskeleton. The cytoskeleton is a protein filament found in eukaryotic cells that extends throughout the cell to serve a variety of functions, such as mobility, motility, the formation of mitosis and cytokinesis, and upholding the overall structural integrity of the cell. The cytoskeleton mediates the mechanical interactions in the cellular environment by organizing the organelle's location, carrying out coordinated movements, and changing shape in response to cellular signals. Pictured here in glowing green and red are the projections of the cytoskeleton that span the cytoplasm of the cell. It is amazing how these proteins arrange themselves in every single one of our cells over and over and over again through cell division. And it is often said that shape implies function in any being, and it is clear from this image that the structure of the cytoskeletal proteins contributes to the many functions we are about to discuss. There are 3 distinct types of cytoskeletal proteins that each serve specialized functions. Pictured here are the basic structures of microfilaments, intermediate filaments, and microtubules, which are pictured on the right as they are actually observed under a microscope. While they do not necessarily seem all that different from one another when we're looking at their basic structures on the left, from the images on the right we can see that these proteins are actually very unique in their localized arrangements. The individual proteins unique to each type of skeletal fiber come together into long, and I mean long, strands of protein fibers to create their complexes. Intermediate filaments specialize in providing the tensile strength for cells in order to counteract mechanical cellular stress. Intermediate filaments form rope-like structures. As the rope that we create is designed to have immense tensile strength, these filaments bundle together and twist around each other in order to create that tension. Pictured here are protein dimers that have heads and tails domains that connect in order to create tetramers, and then protofilaments, which then creates the rope-like structure, imagine this twisted, called an intermediate filament. These filaments are all over the cytoplasm, surrounding the nucleus as the nuclear lamina and extending outwards towards the cell membrane. Intermediate filaments distribute the added weight or stress within the cell like a net in order to avoid tearing from within. Intermediate filaments in particular can be made of keratin, a protein we usually associate with expensive hair products, which not the hair products, but the keratin is often found in our epithelial cells. In the epithelia, the intermediate filaments serve as a variety of functions, and the most important is the withstanding of the mechanical stress on the skin and the lining of the gut as it distends. Vimentin intermediate filaments are found in glial cells, which are the support cells for the nervous system, and in connective tissues. Neurofilaments, another type of intermediate filament, structurally support neurons and aid in its peripheral neurogeneration. And finally, the nuclear lamina is comprised of intermediate filaments that maintain the integrity of the nucleus within the cell, and these filaments can assemble and disassemble in coordination with cell division as the nucleus breaks down and then reforms around itself. Microtubules serve to mediate cell transportation rather than structure. These are long, stiff, hollow tubes that extend out from a centrosome to the phospholipid bilayer, the cell membrane. The centrosome is made of the protein gamma-tubulin that forms a ring-shaped nucleation site where the microtubules begin to arrange themselves from the centrosome outwards. Tubulin is a protein that comprises both the nucleation site and the microtubules themselves, and tubulin can actually be arranged as dimers, as we can see here. There's alpha-tubulin and beta-tubulin, both which are subunits of tubulin that arrange into the walls of the microtubules or protofilaments by stacking non-covalently, and then they form a hollow tube. The microtubules themselves actually exhibit polarity, meaning that a microtubule will always form from the minus end to the plus end, starting with the gamma and alpha tubulin here at the centrosome and ending with the beta tubulin here at the plus end of the microtubule. This gives the cell transportation system directionality, and this dictates the movement of walking transport proteins along the microtubules. Walking proteins can walk along the tubes with transport vesicles in order to move cargo along them. The polarity, directionality, and formation of the microtubules dictates that vesicular transportation through the use of the proteins kinesin, pictured here, and dynein. Kinesins move to the plus end of a microtubule, or towards the cell membrane and away from the centrosome, and interrograde, as we call it. And dyneins move actually to the minus end of a microtubule, and thus towards the centrosome through the retrograde transport system. These proteins literally walk along microtubules, lifting their feet as they cleave ATP through hydrolysis, and swinging one step forward as they release ADP, placing a foot down as they bind and cleave again, literally walking with each step, with each release of energy. I think that this process is just absolutely astounding and beautiful in its simplicity, as the cellular process of transportation along a microtubule is both very simple, complicated, and essential all at once. Microtubules exhibit something called dynamic instability, which means that they go through a sequence of doing and undoing themselves in order to shrink and grow with the transportation needs of the cell itself. Meaning to say that the microtubule is never just static. It's never just built and left there. It's accomplished through the binding of dimers to GDP or GTP as a microtubule is made from plus to minus end. The binding of GDP to a dimer results in the stabilization of the microtubule as it is polymerized, as you can see here. As GDP is cleaved, a microtubule is depolymerized, meaning that it shrinks and undoes itself in response to that change. Being dynamically unstable actually ensures that the microtubules can build endless means for their walking proteins to get the correct cargo to the correct places. Without it, polarity and the composition of tubulin dimers building microtubules, we would be devoid of all of our cellular highways in our cells. Actin protein is essential for cellular movement. Microfilaments or actin filaments are dynamic and unstable. They rely on the binding of several other proteins to maintain its structure and function. The primary function of actin that we will be discussing is contraction of the cell, or movement of the cell. Actin is twisted into polar chains of globular protein subunits that comprise the filaments seen here. These filaments are thin, flexible, and even short when you compare them to their microtubule counterparts. But like microtubules, actin can disassemble easily and assemble itself easily. All actin is bound to ATP when it is cleaved into ATP in order to incorporate actin into a filament through polymerization. This is a costly but essential way to ensure that actin can be dynamic during muscle contraction or movement. Actin filaments rely on many proteins that are highly concentrated along the inner face of the cell membrane, as we see here, where actin binding proteins can give strength and control to the changing of the shape of a cell. Cells can actually crawl or pull themselves in order to migrate and respond to its environment, and this is accomplished through the pushing out of protrusions from the cell. Actin ultimately interacts with another protein, myosin, which we can see here, in order to contract and create movement. The myosin protein family includes all actin binding proteins that bind and cleave ATP in order to accomplish the contraction. Myosins move from the minus end to the plus end of an active filament. Myosin has a myosin head which binds to actin once it's exposed on the troponin and tropomyosin molecule. And the movement of the tail of myosin directs the pulling of the myosin along the actin filament. Myosin moves its lever arm in response to ATP hydrolysis by bending itself. And through the action of binding, pulling, and releasing, microfilaments can perform their function hundreds of times over, all at once. So just to wrap up our conversation on cytoskeletal proteins, here is a table that demonstrates all the differences between the filaments that we discussed. Starting with intermediate filaments, which are composed of IF dimers that form into tetramers, protofilaments, and then the interfilaments we see are so important for the structural support of the cell, but have no known polarity when compared to microfilaments and microtubules, which polymerize from the minus end to the plus end in order to stabilize themselves and to ensure that they can function. Their functions are very different from one another, meaning that microfilaments can change shape and direct the movement in order to make sure that the cell can migrate itself or that muscles can contract, whereas microtubules are important for the cellular highways in our systems by means of transporting molecules down them. Each, although three of these are very, very different, are very, very important to our cellular function overall. Epithelial cells are clustered cell sheets of many types that hold together in stratified layers in order to maintain organization and attachment of lining in our tissues. These epithelial tissues can be barriers. They can secrete or absorb and line our organs. They equate to a cell membrane in a cell for our tissues, organs, and systems. The apical surface of epithelial face outwards, while the basal surface faces inward and rests on the connective tissue, where it connects the basal lamina, a collagen tissue that is known as type 4 collagen. Collagen is the protein that comprises bone, skin, and tendon. Collagen is what makes up the extracellular matrix in which cellular communities exist. Extracellular matrix fibers are arranged based on the needs of the tissues that it exists in, such as in layers, in parallel bundles, or in opposing directions, depending on the function of the tissues involved. There is a range of durability and flexibility of connective tissue that is given by collagen content. Epithelial sheets are polarized based on their apical and basal sides in order to ensure that functions of those areas differ for the tissues, such as absorption, digestion, or adhesion, if you're talking about the lining of the gut or the lining of the skin. Epithelial cells connect to one another and maintain their cellular communities by forming connections or cell junctions. Pictured here are the 4 cell junctions that we will actually be discussing today in great detail: tight junctions, adherence junctions, desmosomes, hemidesmosomes, and a little bit different is the gap junction. Tight junctions are formed by the interaction between claudins and occludin proteins that zip to seal 2 cells together. These seals define membrane domains where the transfer of molecules can occur at the membranes in close proximity. So this is a tight junction here. Claudin proteins and occludin proteins interact in order to pull cells closer together, zip them up, so that way the 2 cell membranes can interact closely in this area. Adherence junctions are junctions that involve cadherin proteins that are tethered to actin filaments in order to create a bond with the cytoskeleton while also bonding the cytoplasm of one cell to another. So here we see 2 cadherin proteins interacting between 2 cells, and the cadherin proteins actually have the same charge, so you would think that they would repel one another. But there's actually a calcium that binds the 2 cadherins together here in order to stabilize the cell junction. And this cell junction is pivotal in maintaining the integrity of the cellular community by reinforcing the mechanical strength in the cell across the length of several cells, because cadherins here are actually attached to the actin filaments. When a cell is going through cellular stress, those actin filaments will rely on one another across their cells in order to maintain their structure. Desmosomes are actually quite similar to adherence junctions, as they actually also utilize cadherin proteins to create a bond across the cells through the joining of intermediate filaments, or keratin, instead of actin. Desmosomes are also responsible for maintaining the mechanical strength of the epithelial cell layer. So instead of connecting actin between these cells, it is the intermediate filaments that are connected instead, and this just reinforces the cell's response to the mechanical stress that it experiences. In hemidesmosomes, integrins link to collagen in order to form a bond between the basal lamina and the basal epithelial regions. This is essential to cellular communities, especially epithelial cells, in order to ensure that there is no leakage of particles between the apical side of the epithelial and the basal side. Hemidesmosomes help to form a tight, impenetrable layer so that the skin does not just slide off of our bodies, which would be quite strange. So as you can see, these are the desmosomes here we were talking about with the intermediate filaments. And hemidesmosomes connect to the basal lamina to the extracellular matrix and the connective tissue in order to ensure that the epithelial cells stay put here on this layer. Connexon transmembrane proteins can create gap junctions between cells in order to create openings between cells for small molecules to pass through for private communication between those cells. Cells that have gap junctions are electrically and chemically coupled and regulated in this way. Gap junctions are actually often associated with neurons, my favorite cell. as neurons need to be electrically and chemically coupled by gap junctions or synaptic transmission in order to accomplish the amazing things that it does. So here you can see closely a synapse between two neurons where there is a gap junction. Individual connexins make up a connexon, which is like a pore that forms between the two membranes, so that any of the ionic changes that carry over from one neuron will just carry over to the next instantaneously. And this actually, fun fact, is not very common in the central nervous system of humans. Synaptic transmission is the mode of communication usually between neurons, but this electric coupling between the two neurons can be useful in certain places. So just to review all 5, I accidentally mentioned 4 when I introduced all these, but all 5 of these connections and their importance to maintain the structure of our cellular communities. Tight junctions are really important to prevent the leakage between cells because they're so tightly zipped together. It ensures that nothing really gets through. Adherence junctions are important for structural stability of the cell, and desmosomes are important for the stretching of the cell against mechanical stress. Hemidesmosomes are important for watertight binding to the basal lamina, and gap junctions are important for the communication and coupling of two cells when needed. And without all of these cell junctions in certain places of our bodies, our cells would not behave the way that they do to ensure that we are sustained as a larger living multi-system of cells. So cancer is a topic that hits pretty close to home for me, as I've had my own experience with cancer itself, as many other people have in their lives. Cancer is a unique disease because there is difficulty in fighting it due to its inherent role within organisms. It's not caused by pathogens, but instead by the mutations within an organism's cells. Because we live long lives due to the cell cycle and our production of stem cells, there are consequences to our ability to repair, proliferate, divide, and differentiate into healthy cell systems over time, and it changes, it degrades. Cancer is defined as a disease of erroneous cell behavior and breakdown over time, and that is a major cause of death on this earth. Cancer can be studied in every field of biology, as it violates many aspects of normal order of cell structure, function, and life. Pictured here are actually microscopic images of cervical cancer cells from a human mouth, which was measured and analyzed using a colored scanning electron micrograph. As you can see, these cancer cells actually have motile projections, which is really an interesting feature of a cell that is invasive. Cancer gets to break all of the rules that cells follow in order to maintain a coordinated, highly regulated, complex system of cells based on signal response to the organism and to one another to maintain that balance. Cancer is characterized by a mass of cells that break these rules continuously and continue to divide to pass these terrible qualities along. And this compromises the organism's ability to function and survive normally. Cancer is specifically defined by its ability to proliferate and to invade and colonize in areas where other cell types reside. If cells continue to proliferate but not invade, they are considered benign tumors. If cells meet both definitions, then it is malignant. If cancer cells have secured a blood supply, it can do the above-mentioned things and cause significant harm through the spreading, and these are the metastatic tumors that spread to the rest of the body, which give rise to secondary tumors through the circulatory system. These tumors are much harder to battle in an organism. This is because cancer cells do not need extracellular signals in order to survive. They do not go through programmed cell death, are invasive, undying, and genetically unstable. Cancer is a disease of cellular mutations, meaning that it exclusively results from somatic mutations that are caused spontaneously throughout our cells. Cancer is caused by the accumulation of these random mutations where genetic instability is directly related to the increased likelihood of cancer. Mutations that directly cause defects in DNA repair or cell cycle checkpoint regulation can cause the extreme buildup of spontaneous somatic mutations. The danger of cancer cells is that they are in competition with normal cell function and can easily outcompete normal cells due to its rule breaking. They can also survive in cell regions among specialized cells they are not meant to associate with. Oncogenes are genes that can become hyperactive when they are dominantly mutated, meaning that only one of them needs to be mutated in order for the mutation to appear in an organism. And an unmutated oncogene is called the proto-oncogene. Oncogenes code for rapid cell division during development, and so they are turned off when they are not mutated. So when they are mutated, cells are rapidly dividing when they should not be. Tumor suppressor genes, when recessively mutated, meaning that both of the alleles for the mutation must be there, are turned off when they are actually not supposed to be downregulated. A genetic predisposition to cancer is determined by the recessive allele that are tumor suppressor genes. Most tumors are actually affected by both tumor suppressor genes and oncogenes. It is not important what types of mutations are occurring, but instead where those mutations occur. Gene mutations that result in cancer occur in genes that control cell growth and proliferation, and responses to cellular stress and DNA damage can ultimately be impacted by these mutations. Hopefully, there are advancements in treatment that can help us approach these issues in order to stop cancer before it truly starts. I hope you enjoyed my eReport. Thank you so much for joining me for these 35 minutes.